Welcome to AP Biology. This video is on microbial genetics, the genetics of bacteria and viruses. Here we see a picture of a eukaryotic animal cell, a bacteria, and a virus. Remember that the animal cell is eukaryotic. You can see that it contains a nucleus. It contains membrane-bound organelles. The bacteria is prokaryotic. It's much smaller than the animal cell. We know that bacteria lack a nucleus. Instead, they have a general region called the nucleoid region. Here you can see the single circular chromosome inside the nucleoid region. The bacteria also contains no membrane-bound organelles. If you look at protein synthesis in a prokaryotic cell versus a eukaryotic cell, you can see that protein synthesis is much more simple. There are no introns and exons in the prokaryotic DNA. There's no nucleus for transcription to take place in. When the DNA gets transcribed into messenger RNA, the messenger RNA does not receive any processing. Instead, it goes right to a ribosome where the messenger RNA will be translated into a protein. Both eukaryotic cells and prokaryotic cells, they're both considered living because they are made up of cells. The virus, on the other hand, is not considered alive because it is not made up of a cell. Instead, viruses are made up of genetic material surrounded by a protein coat. You can see that the virus is much smaller than even the bacteria. Here you can see 4 different types of viruses. We see the tobacco mosaic virus, the adenovirus, the influenza virus, and the bacterial phage. Each virus is made up of genetic information. That genetic information could be DNA. It could also be RNA. Surrounding the genetic information, we have a protein coat. The protein coat, you see right here, is called the capsid, and the capsid is made up of proteins called capsomeres. You can see that each virus has its own characteristic shape. You can see that the tobacco mosaic virus looks like a cylinder, and its genetic information is made up of RNA. Here you see the adenovirus. This is the type of virus that causes the common cold, and its genetic material is made up of DNA. The influenza virus is the virus that can cause the flu, and you can see it has its own spherical shape, and its genetic information is made up of RNA. Finally, here we see the bacteriophage. The bacteriophage looks like a tiny spaceship, and its genetic material is made up of DNA. We'll see the bacteriophage more in a little bit. The bacteriophage is a type of virus that only infects bacteria. Some viruses also have another structure. It's called the viral envelope. Here you can see what a viral envelope looks like. It's made up of glycoproteins and membrane. The viral envelope is made up of a piece of membrane from the host cell. That way the virus can enter the host cell through endocytosis, and it could leave the host cell through exocytosis. A virus is a true parasite. It lacks all enzymes, it lacks all ribosomes. It literally uses the host cell against itself in order for the virus to reproduce. Every virus has a specific host range. The host range is the particular type of cell that the virus infects. For instance, the HIV virus only infects helper T cells, which are white blood cells found within the immune system. Here you see how a virus uses the host cell to reproduce. A virus is going to enter the host cell through a lock and key mechanism. In particular, the genetic information is going to enter the host cell. Once the genetic information enters the host cell, it's going to use the host cell's enzymes and the host cell's ribosomes in order to first replicate all the viral genetic information and second, to make all the viral proteins, those capsomeres. Once the genetic information has been replicated, once the viral proteins have been created, the viruses will self-assemble and exit the host cell. In the process of exiting the host cell is usually killed as an end result. Here we see a short animation showing us how viral reproduction takes place.
A virus is an intracellular parasite that can reproduce only by taking over a host cell. A virus consists of a nucleic acid genome enclosed in a protein shell called the capsid. In the virus shown here, the genome consists of DNA, but some viruses have RNA. Some viruses are also covered by a membranous envelope that is derived from the membrane of the host cell. There is usually a lock and key fit between the proteins of the capsid and receptors on a particular type of host cell. The virus attaches to a host cell and viral DNA enters the cell. The viral DNA uses nucleotides and enzymes of the host cell to replicate itself. The viral DNA then commandeers other host cell materials and machinery to transcribe its genes into messenger RNA and translate the RNA message into capsid proteins. Viral DNA and capsid proteins then assemble into new viruses. Mature viruses leave the host cell, often destroying the cell in the process. The viruses can go on to infect other cells, spreading the viral infection.
Here we see a scanning electron microscope picture of bacteriophages infecting the host bacteria. There's two types of reproductive cycles that a bacteriophage or a phage can enter. One is called the lytic cycle. The second is called the lysogenic cycle. The lytic cycle is ultimately going to kill the host bacteria. Virulent viruses are viruses that undergo the lytic cycle. When the bacteriophage lands on the host bacteria, in this case the host bacteria is E. coli, it's going to insert its DNA. Once that DNA has been inserted, the host cell's DNA gets cut up. It's going to get cut up through the process of hydrolysis. The bacteriophage then uses the host cell's enzymes and the host cell's ribosomes to manufacture new bacteriophages. Those bacteriophages will self-assemble and will break out of the host cell. You can see in this picture here that when the bacteriophages exit the host cell is going to die in the process. Again, here we see a short animation showing how the lytic cycle takes place.
Bacteriophages, or phages, are viruses that infect bacteria. This is a T4 phage, which consists of DNA inside a protein coat. The lytic cycle begins when the tail fibers of the phage stick to receptor sites on the surface of a host bacterium, such as E. coli. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The DNA of the host cell is destroyed, and host cell enzymes and nucleotides are commandeered to replicate the phage DNA, making more phage DNA. The host cell's enzymes and ribosomes transcribe the phage genes and translate them into phage proteins. Phage parts accumulate and assemble to form phages. A phage enzyme digests the bacterial cell wall and the cell ruptures or lyses. As many as 200 phages spill out. Each of them may go on to infect another cell. This diagram summarizes the lytic cycle of bacteriophage T4.
The other reproductive cycle that a phage virus can use is called the lysogenic cycle. The lysogenic cycle is not going to kill the host cell. Viruses that reproduce using the lysogenic cycle are called temperate viruses. Here you can see the phage infecting the host bacteria. Once the phage inserts the genetic information, that DNA is then going to incorporate itself into the host cell's DNA. Once the virus's DNA has been incorporated into the host cell's DNA, we now call it a prophage. Once that prophage is formed, every time the bacteria reproduces, it's also going to reproduce viral DNA. Every time the bacteria produces proteins, it's also going to produce viral proteins. Eventually, that prophage will leave the host cell's genome. Once that prophage leaves the host cell's genome, the virus will then convert from the lysogenic cycle to the lytic cycle, which again is ultimately going to kill the host cell. Here we see an animation showing the lysogenic cycle.
In contrast to the lytic cycle, the lysogenic cycle reproduces the viral genetic material without destroying the host. The lysogenic cycle of phage lambda begins when a phage binds to the surface of a host bacterium. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The viral DNA is incorporated into the host cell DNA, where it is called a prophage. Every time the host bacterium reproduces, it replicates the phage DNA along with its own and passes the copies on to daughter cells. Occasionally, the phage DNA exits the bacterial chromosome and initiates a lytic cycle. The viral DNA takes over the metabolic machinery of the host cell to make phage DNA and proteins. The host cell lyses, releasing phages which can infect other cells. This diagram summarizes the lysogenic and lytic cycles of phage lambda.
Here we see a comparison of the lytic and the lysogenic cycle. You can see the key difference is that the lytic cycle is going to kill the host cell, where the lysogenic cycle does not directly destroy the host cell. Here we see the HIV virus. The HIV virus is a retrovirus. It's called a retrovirus because it has RNA as its genetic information. If you look at the virus itself, here you could see the capsid, or the protein coat. Inside the capsid, we see two strands of ssRNA, single-stranded RNA. Inside the capsid, along with the RNA, is an enzyme called reverse transcriptase. Reverse transcriptase is an enzyme that's going to perform transcription in reverse. So it's going to take the RNA from inside the virus, and it's going to be able to convert it into DNA. Surrounding the capsid, we can see the viral envelope. And again, that viral envelope is made up of pieces of the host cell's membrane. So if we look at this picture here, you can see that the HIV virus can enter the white blood cell through endocytosis, and it can exit the white blood cell through exocytosis. The particular type of white blood cell that HIV infects is called the helper T cell. Let's look at what happens. The HIV virus enters the host cell through endocytosis. Once the viral RNA has entered the host cell, reverse transcriptase is going to reverse transcribe the viral RNA into DNA. Once that DNA has been created, it's going to enter the host cell's genome and become what we call a provirus. A provirus is the same thing as a prophage, except that prophages are found in bacteria, where proviruses are found in eukaryotic cells. Now, every time the white blood cell goes through protein synthesis, it's going to manufacture new RNA. That RNA is the genetic material for the HIV virus, and it's going to manufacture new viral proteins. At some point, the HIV viruses will self-assemble inside the white blood cell, and they will exit the white blood cell through exocytosis. When exocytosis takes place, the viral envelope will be formed. When these viruses exit the host cell is going to be destroyed as an end result. Here we see an animation showing us how HIV virus infects white blood cells.
Understanding the structure and life cycle of HIV, the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studying the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of two molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
To prevent a viral infection, an individual might receive a vaccination. A vaccination is going to consist of either viral proteins, viral genetic information, a denatured heat-killed virus, or other sources. The goal of a vaccination is for the individual to build up an immune system response for the virus. So the individual is going to create antibodies as a result of injecting a small piece of that virus into the individual. That'll help protect that individual if they ever encounter that virus in the future. Besides viruses, there are other simple infectious agents. Viroids are circular pieces of RNA that can infect plants. Prions are infectious proteins. We learned in the past that prions are misfolded proteins, and they cause the proteins within the body to not properly function. An example of a disease caused by a prion is called mad cow disease. Now we transition from viruses to bacteria. Remember, bacteria are small prokaryotic organisms. You can see here they lack a nucleus. Instead, their single circular chromosome is found in a general region called the nucleoid region. They also lack all membrane-bound organelles. The only organelle you'll really see inside a bacteria is going to be the ribosome. Besides having the single circular chromosome, bacteria have extra small pieces of DNA called plasmids. Plasmids are small circular pieces of DNA that can contain anywhere from 1 to a few genes. These plasmids are going to be very important in terms of genetic recombination that takes place in bacteria. We'll find that bacteria have the ability to transfer these plasmids from one bacteria to another bacteria, and it's a way to mix up their genes. It's a way to create genetic variation. Bacteria reproduce very quickly. They have a short generational span. They reproduce through binary fission. Binary fission, we know, is a form of asexual reproduction. Since they reproduce asexually, they need a way to create genetic variation. Genetic variation is crucial for the process of natural selection, the method by which evolution takes place. Bacteria have an extremely high rate of mutation, which is going to lead to lots and lots of genetic variation taking place. Besides the mutation rate that leads to genetic variation, there are 3 forms of genetic recombination that take place inside bacteria. Here you see an investigation that shows genetic recombination taking place. We see bacteria grown in the absence of particular genes. When bacteria grows on a petri dish, they are going to reproduce and form what we call colonies. Colonies are made up of thousands of bacteria. Here we see one strain of bacteria grown on a petri dish. They contain the gene to create arginine, but they lack the gene to create tryptophan. When they lack the gene to create tryptophan, no colonies will form. Here you see another strain of bacteria. They have the gene for tryptophan, but they lack the gene for arginine. When they lack the gene for arginine, they also cannot reproduce and form colonies. However, when you put these two strains together, you can see that colonies do form. In order for those colonies to form, they need to have the genes for both arginine and tryptophan. So in order for this to take place, genetic information must have been transferred between those two strains of bacteria. Here we see three different methods by which that genetic information can be exchanged. One is through the process of transformation. Transformation is when a bacteria takes in plasmids from their environment. In order to take in a plasmid from the environment, we must induce what we call competency in the bacteria. In our lab, we are going to induce competency in bacteria through a process called heat shocking. Heat shocking is when you expose bacteria to a high temperature and then to a low temperature. When you do this, it opens up specialized surface proteins on the cell membrane that allow the bacteria to take in these plasmids. Once the bacteria has taken in these plasmids, they can express the genes on the plasmids and they'll have a new trait. That trait might allow them to do something like be resistant to antibiotics. Another way that bacteria transfer information is through a process called transduction. Transduction is when phages are used to transfer DNA from one bacteria to another bacteria. This occurs accidentally. There are two forms of transduction. We have generalized transduction and specialized transduction. For the most part, these two processes are the same. The only difference is that in generalized transduction, the phage is going to utilize the lytic cycle. In specialized transduction, the phage is going to utilize the lysogenic cycle. In either case, with transduction, the phage accidentally transfers DNA from one bacteria to another. Here we see on the left how generalized transduction takes place. The phage is going to land on the bacteria. You can see that it inserts its genetic information. The bacteria's DNA is cut up. New phages are created and they leave the host cell. However, you can see right here though, inside one of those phages, instead of viral DNA being found inside that phage, bacterial DNA is found inside instead. Now when this phage exits the host bacteria and it infects a new bacteria, instead of injecting its own viral genetic information, it's injecting the DNA that came from the prior bacteria. So you can see here a mistake allows bacterial DNA to be transferred from one bacteria to the next. In specialized transduction, essentially the same thing is going to take place, except that the phage is going to utilize the lysogenic cycle. Here you can see in the lysogenic cycle that the prophage is going to leave the bacteria's DNA. Except you see right here a mistake takes place. Inside that prophage we have a small piece of bacterial DNA. So when new viruses assemble, inside one of those viruses is going to be bacterial DNA. And when that virus infects a new bacteria, it's going to insert the DNA from the prior bacteria. Here you see the third method by which DNA can be transferred from one bacteria to another bacteria. We call this conjugation. This is most likely what took place in the experiment that we looked at before. Conjugation is also called bacterial mating. Some bacteria have a plasmid called the F plasmid. Here you see what the F plasmid looks like. F stands for fertility. The F plasmid contains genes that allow the bacteria to form what we call a sex pili. A sex pili is a projection that sticks off of the bacteria, and it allows the bacteria to form a cytoplasmic bridge with another bacteria. Once that cytoplasmic bridge forms, that plasmid can then be replicated and move to the second bacteria. Once that bacteria receives the F plasmid, it then has the ability to now create a sex pili for itself. When a bacteria receives the F plasmid, that F plasmid is going to insert itself into the bacteria's DNA. Here you can see it taking place in the middle. Once that F plasmid has inserted itself into the bacteria's DNA, that bacteria then gets referred to as an HFR cell. HFR refers to a high frequency of recombination. It's called an HFR cell because it can now actively form cytoplasmic bridges with other bacteria and actively exchange genetic information. Bacteria that have the F plasmid are referred to as male bacteria because they can create a sex pili. Bacteria that do not have the F plasmid are referred to as female bacteria because they cannot form the sex pili. Another plasmid that we're going to see exchanged between bacteria that's of particular importance is a plasmid called the R plasmid. The R plasmid contains genes that allow the bacteria to become resistant to antibiotics. So here you saw the three methods by which genetic information can be directly transferred from one bacteria to another bacteria. Transformation is the direct uptake of plasmids from the environment. Transduction is when a phage accidentally transfers DNA from one bacteria to the next. And conjugation is going to be bacterial mating, when bacteria directly transfer genes from one to the other using a cytoplasmic bridge. That F plasmid contains the genes that will allow the bacteria to form the cytoplasmic bridge, and these bacteria that have the F plasmid inserted into their genome are referred to as HFR bacteria, bacteria that have a high frequency of recombination. These three methods, along with a high rate of mutation, they allow for tons of genetic variation in bacteria. Genetic variation is at the heart of Darwin's theory of natural selection, and that's going to allow these bacteria to evolve very quickly. Finally, within the genome of both prokaryotic organisms and eukaryotic organisms are transposable elements. Transposable elements are commonly called jumping genes. They're called jumping genes because they have the ability to move from one part of the genome to another part of the genome. They don't actually jump, instead they use a cut and paste mechanism using nucleases and ligases. In bacteria we see insertion sequences. Insertion sequences contain only a single gene that has the ability to move. Transposons are found more in eukaryotic organisms. They're longer and they're more complex sequences. However, again, they are able to move from one part of the genome to another part of the genome. These transposable elements, these jumping genes, increase the amount of genetic variation in both bacteria and eukaryotic organisms. That concludes this video. Thank you.